Very good morning to all the students. So, today we are going to start our bridging course. Firstly, we will begin with the chapter Cell the Unit of Life. Why we are starting with this chapter? Because once we understand the cell structure of both prokaryotic cell, eukaryotic cell, of plant cell and animal cell, and after having complete idea about all the organelles and its functions, we will get to know the further concepts more detailedly, like animal physiology, plant physiology, structural organization of animals, be it plant kingdom, morphology, anatomy. Everything will be clear to you once you get proper idea on the cell. So, that is why we are beginning with this chapter. So, in this chapter, what are the contents we have to cover with respect to the NEET examination? Firstly, the fundamental insights of this chapter includes, first we will learn about cell and cell theory, its discoveries, and then the types of cells. We have both prokaryotic cell, eukaryotic cell. In that prokaryotic cells also you will study the variation of size and shapes with respect to the diagrams, and eukaryotic cells also you will learn from unicellular to multicellular cells, different shapes of the cells as well. And then you will learn about the structures. Coming to the cell wall, what will be the cell wall organization of plant cell, of algal cell, of fungal cell, and you will learn about the cell membrane. What is the widely accepted model of the cell membrane? Fluid mosaic model given by Singer and Nicholson. And then you will learn about cell organelles. Here we are going to learn cell organelles in a different pattern by organizing or grouping them into amembranous cell organelles, single membrane bound cell organelles, or double membrane bound cell organelles. So, in these three patterns we are going to learn all the cell organelles. When we are learning single membrane bound cell organelles, there comes the concept of endomembrane system. And then double membrane bound organelles, name few double membrane bound organelles: nucleus, mitochondria, chloroplast. These are the double membrane bound organelles. Last but not the least, it is about cytoskeleton, where we are going to learn about microtubules, microfilaments and intermediary filaments. Along with this, under amembrane bound organelles, we are going to learn about centrioles as well. So, these are all the contents of this chapter, where the highest proximity of questions from where you will see, mostly from prokaryotic cell, okay. Either it can be from fluid mosaic model or from cell theory, and there will be a definite question from endomembrane system, double membrane bound organelles, specially about the types of chromosomes. So, from these areas we have seen the highest proximity of the questions. In this class we are going to learn bridging details from your foundation to your intermediary, wherein clearly we are going to learn about the cell theory, different types of cells, shapes of cells, prokaryotic cell, eukaryotic cell, plant cell and animal cell. And we are going to solve 5 questions and few of the previous NEET questions just on this class itself. See how easy it will be. First, about the cell. The solid statement that is given for a cell is what? Cell is the fundamental, structural and functional unit of life. Why is cell called as fundamental, structural or functional unit of life? Why is it called like that? Because a single cell is independent in its function, yes or no? It is independent in its function, and it is independent in its structure as well. As such, if you look into the hierarchy of organization, group of cells will form what? Tissues. They can be similar or dissimilar in structure, but they are functionally similar. Structural similarity might be there or not, but functionally they are similar. They will form a tissue, because definition of tissue is very much important. Quite a times you got statement based question. Group of cells which can be structurally similar or dissimilar, but functionally they are similar, those form a tissue. Group of tissues will form organs, organ systems and then organism. As such, the hierarchy of living organization will move, fine. and cell is structurally and functionally independent, so it is called as structural and functional unit of life. And the cell is capable of independent existence and it is performing all the essential functions on its own. Now, if we have to look into few discoveries, see here in 1665 Robert Hooke has discovered what? Dead cell, okay, in cork cells of Quercus suber. So, from this plant, from the bark, he has identified the cork cells like this, which are to be like a honeycomb structure. Okay. Why is it called as dead cell? Why is it called as dead cell? It is because there is no protoplasm in it. Okay. It is because there is no protoplasm in it, that is why it is called as dead cell. So, who identified the dead cell first? Robert Hooke. In which year? 1665. From what cells? Cork cells. Fine. Next, in 1674 it is Anton van Leeuwenhoek who has discovered the living cells, those which has protoplasm. Next, who discovered the nucleus? Nucleus is discovered by Robert Brown, and in 1838-1839 two scientists, one is Matthias Schliden, he is a German botanist. Who is he? German botanist. And Theodore Schwann, who is he? British zoologist. So, Matthias Schliden, German botanist, and Theodore Schwann together proposed what ma? Cell theory. And then in 1885, one scientist has modified the cell theory. He is Rudolf Virchow. We are clear with the basic discoveries of who discovered dead cell, who discovered the living cell, and who gave us the nucleus. Now, we will discuss in detail about cell theory and objections to cell theory or exceptions to cell theory as well. Cell theory, as we have discussed, Matthias Schliden, he is a German botanist, he said only one point beta, that all plants are composed of cells, that is it, okay, and cells are the products of cells. Whichever cells are present, they are the products of cells, he said. Only 2 points. What are they? All plants are composed of cells, cells are the products of cells. And next, Theodore Schwann in the year 1839, he said that he has observed both plant cells and animal cells. Though he is a British zoologist, he is the person who said cell wall is the limiting boundary for a plant cell. Cell wall is the limiting boundary for a plant cell, is said by whom? Theodore Schwann, though he is a British zoologist. And he said the limiting boundary in an animal cell is what? Cell membrane. Cell membrane is the limiting boundary of an animal cell. Fine, understood? So, who gave us the cell wall is, who discovered the cell wall? Schwann. And who gave us the cell membrane is the outer membrane of the animal cell? Schwann. He has observed both plant cells and animal cells. But both of the scientists have failed to say about the newly formed cells. How new cells are formed, both of them didn't explain. In the year 1855, the year which year ma? 1855, Rudolf Virchow has said that new cells arise from the pre-existing cells. Already those cells which are existing, they undergo division and they will give rise to new cells. So, new cells arise from pre-existing cells. This is called as omnis cellula. Very good. Omnis cellula, E cellulae. Fine. So, now we are clear with this. Cell theory is applicable to what? Cells. Okay. What type of cells? I will tell you. One cell, okay, which has nucleus in it, cytoplasm. Nucleus and cytoplasm together it is called as protoplasm, and it should have a cell membrane. So, cell theory is applicable to what cells, Nana? The cells which has nucleus, cytoplasm, cell membrane. Ivi moodu undali, then only it is applicable, cell theory is applicable to those cells. Okay? So, nucleus and cytoplasm together is called as what? Protoplasm. Along with cell membrane it is called as what? Protoplast. So, this is clear to your mind now? Cell theory is applicable to those cells which has nucleus, cytoplasm and a cell membrane. There is any thing that is missing out from the cell, then cell theory is not applicable. So, now I will tell you, see, cell theory is not applicable to. First question, is it applicable to those cells which does not have nucleus? Is cell theory applicable to the cells which does not have nucleus? No, because cell theory is applicable to only these cells, right beta? Cell membrane, nucleus and cytoplasm. If more untane cell theory is applicable. Nucleus is not there, means cell theory is applicable? No. So, here tell me few cells in which nucleus is absent. Sieve tube cells. Sieve tube cells in plant inside the phloem, fine. To this nucleus is absent. Tell me one more type of cell, matured RBC, nucleus is absent. Yes, mature RBC. And now one more question, if in a cell there is more than one nucleus, is cell theory applicable? No. Okay. So, second point, more than one nucleus. You can see it in some protozoans like paramecium and amoeba has only one nucleus beta. And think, first is like 2 nucleus condition in paramecium, you can see. Rhizopus, bread mold. Okay. So, here there is more than one nucleus. To this also cell theory is not applicable. To living entities like viruses, they does not have cell, they just has a genetic material and a protein coat like this. To viruses also cell theory is not applicable, fine. So, what are the exceptions of cell theory, ma? Those cells which does not have nucleus like sieve tube cells and mature RBC, those cells which has more than one nucleus like paramecium protozoans or bread mold like rhizopus, those cells which does not have cell membrane or cytoplasm, they just has genetic material and a protein coat, for example virus. To all these cells, cell theory is not applicable. So, these are what? Exceptions of cell theory. Understood? So, now it is clear to you know what is cell theory, who gave us the cell theory, who gave us the modified cell theory, fine, and what are the exceptions of cell theory. Clear, right beta? Next. Now, we will look into different shapes of the cells, fine. First is red blood cells, these are round and biconcave. White blood cells are amoeboid. Columnar epithelial cells long and narrow, and nerve cell which is branched and long, tracheids elongated, mesophyll cells round and oval. Now, here comes the concept of smallest cell. What is the smallest cell? Mycoplasma is the smallest cell. What is the largest cell? Ostrich egg cell is the largest cell. What is the longest cell? Nerve cell is the longest cell. So, smallest, largest, longest cell, keep it in your mind. Next, here before going into the detailed discussion of prokaryotic cell, I would like to put one simple question to you. So, is cell theory applicable to prokaryotic cell? Is cell theory applicable to prokaryotic cell? Students in online also, you can answer through chat box beta. Yes, nucleus is not there, but in prokaryotic cell, nucleoid is present, right? Nucleoid is present. Genetic material is not completely scattered in the cytoplasm, no ma. It is kept at only one place, you will call it as what? Nucleoid, okay. So, it is at one place, that is nucleoid, and completely it is filled with what? Cytoplasm, and it has a cell membrane. So, cell theory is applicable to prokaryotic cell. Itlanti questions vaste, you should not get confused. Fine. Simple. Cell theory is applicable to those cells which has one nucleus, cytoplasm, cell membrane. So, whichever options are given to you, just look into those cells whether they are having these three or not. Based on that, you can eliminate it. Now, prokaryotic cell. So, yes, first we will start from cell envelope, then we will go to cytoplasm, then we will go to the organelles and genetic material present in it. In a prokaryotic cell, it is bounded by what? Cell envelope. Cell envelope. Fine. Prokaryotic cell has what? Cell envelope. Cell envelope here it is 3 layered, not just 1 layer. It is how many layers? 3 layered. The very first layer is what ma? Capsule. Second is cell wall. Third is cell membrane. Okay? Capsule, cell wall and cell membrane. Sorry, first is what is glycocalyx. First is what? Glycocalyx. Glycocalyx, cell wall and cell membrane. So, Nandu, glycocalyx can be of 2 types ma. It can be either as capsule, it can be either as capsule, or it can be as slime layer. Fine. If glycocalyx composition is very thick and tough, it is very thick and tough like this, then it is called as capsule. Like here you can see the outermost layer is what? Capsule. Glycocalyx can exist as capsule and slime layer. If it is thick, it is called as capsule. If capsule is present as the outer layer, then what do you call it as? How is it helpful to the bacteria? If capsule is at the outer layer, how is it helpful to the bacteria? It will prevent the bacteria from host immune system. From host immune system. And it will prevent the bacteria from the attacks of bacteriophage. You know what is a bacteriophage? Bacteriophage will infect the bacteria. So, if capsule is there as the outer layer, it will prevent the effect of bacteriophage, thereby preventing the infections of bacteria. 2 points clear to you? If capsule is not present, outer layer is made up of slime layer, then how is it helpful to bacteria? It will prevent from the dehydration of bacteria. For example, bacteria is in some halophilic condition, thermoacidophilic condition, fine, that during that conditions this will be prevented, fine. It will prevent the dehydration of bacteria. If it is slime layer, it will prevent the bacteria from dehydration. So, clear for you the outer layer glycocalyx. Next second layer is what ma? Cell wall. Second is what? Cell wall. Cell wall in bacteria is made up of what? Peptidoglycan. Cell wall is made up of peptidoglycan. Just a second. Venkatesh, I will talk to you at the end of the class ma. Yes. So, cell wall is made up of peptidoglycan. Next is cell membrane. So, here the cell membrane of bacteria is little peculiar beta, wherein if you consider this as cell membrane has some extensions. These extensions can be tubular like this, or it can be lamellar like this, or it can be in the form of vesicles. Okay. So, these extensions are called as mesosomes. Very good. It is called as mesosomes. Fine. This can be in the form of what? Tubular, lamellar, or in the form of vesicles. The most prominent statement is mesosomes of prokaryotes are equivalent to mitochondria of eukaryotes. Why? First we have to know the functions of mesosomes. Mesosomes are helpful in cell wall formation, DNA replication in bacteria, respiration and secretion. And as these has these extensions, no, that is increasing the surface area of the cell membrane, thereby increasing the surface area also. So, all these functions are very important, ma, because we have got many questions from the functions of mesosomes itself. Okay. So, what are the mesosomes? They are the cell membrane extensions of prokaryotes towards the cytoplasm. Fine. What are the functions of mesosomes? Cell wall formation, DNA replication, respiration, secretion, increasing the surface area. How is it performing respiration? By releasing out some respiratory enzymes into the cytoplasm. That is why it is also promoting what? Secretion. As it is performing the respiratory activity, mesosomes of prokaryotic cell, you will call it is equivalent to mitochondria of eukaryotic cell. Now, are you clear with the cell envelope, ma? The very outer layer of cell envelope is what? Glycocalyx. It can be in the form of capsule or slime layer. If it is in capsule, what is the function is it doing? Preventing the bacteria from host immune system and from infections of bacteriophages. Clear? If it is in the form of slime layer, what is it doing? It will prevent the bacteria from dehydration. Second, cell wall. Cell wall composition is what? Peptidoglycan, means it has peptides and glycan. Glycan is in the form of NAG units. What units? NAG units. You will call it as N-acetyl glucosamine. What do you call it as, beta? N-acetyl glucosamine. So, here it is in the form of peptidoglycan, and cell wall has one more composition. You will call it as murin, and it is rich in teichoic acids. Cell wall compositions of prokaryotic cell most important. For eubacteria, what do you have? Peptidoglycan. Glycan is in the form of NAG units. It has murin and teichoic acid. Cell membrane has these extensions, what you call it as? Mesosomes, and these are its functions. Clear with the cell envelope? Now, if you have to look into the cytoplasm, the cytoplasm of the bacteria, it has non-membrane bound organelles like ribosomes. Okay, it has non-membrane bound organelles like ribosomes, and first we will discuss about the genetic material, then we will go for the ribosomes. It has two types of DNA. Prokaryotic cell will have how many types of DNA? Two types of DNA. What are they? One is called as chromosomal DNA, and the second one is called as extra chromosomal DNA. What is the chromosomal DNA? It is the actual genetic material that is present in the prokaryotic cell, okay, which is double stranded circular DNA. It is double stranded circular DNA. Extra chromosomal DNA is also called as non-genomic DNA or plasmid DNA. It is called as what, ma? Non-genomic DNA or plasmid DNA. Fine. So, this plasmid DNA, it has some particular functions, unique functions for the prokaryotic cells. Like what? Giving particular phenotypic character to the prokaryotic cell, providing the resistance for antibiotic, okay, promoting antibiotic resistance, and promoting the conjugation, okay, transfer of fertility factor in reproduction, or you can say sexual recombination. What are the three functions of plasmid DNA, ma? Sexual recombination, antibiotic resistance, particular phenotypic character to the bacteria. Remember these three functions for the plasmid DNA. You got clear idea, right? What is the functions of chromosomal DNA and plasmid DNA? Synonyms for plasmid DNA? Extra chromosomal DNA or non-genomic DNA. Now, what are the non-membrane bound organelles? Ribosomes. Ribosomes. So, these ribosomes are around 15 nanometers to 20 nanometers in length, discovered by George Pallade. Where are these ribosomes present in the prokaryotic cell? I will say you see. Few ribosomes are attached to the cell membrane, and few ribosomes are freely present inside the cytoplasm like this. Fine. Each ribosome will have two subunits. What are they, Nana? Large subunit and small subunit. In prokaryotic cells, what type of ribosomes you see? 70S type of ribosomes. Okay, you see 70S type of ribosomes. Here larger subunit is 50S, smaller subunit is 30S. Remember that. And where are they present? What are the locations? Attaching to the plasma membrane or cell membrane, and few are freely present in the cytoplasm. This is the complete information about the prokaryotic cell. Okay. Last and final discussion, that is about the motile and non-motile structures of the bacteria. Okay. What are the motile and non-motile structures, ma? Motile and non-motile. Both these motile and non-motile structures, according to your NCERT, those are cell wall extensions. Fine, both motile and non-motile structures, those are what beta? Cell wall extensions. But the motile structure, only motile structure of prokaryotic cell is flagella. Okay, it is what, ma? Flagella. Cilia is not present in prokaryotes, do remember. And the flagella is having what type of protein? Flagellin. In eukaryotic flagella, the protein which is present is tubulin. Remember that. Here it is what? Flagellin. Okay. Next, what are the non-motile structures present in the over the cell wall extensions? They are pili and fimbriae. What are they, ma? Very good. Pili and fimbriae. Okay. Pili is called as the tubular extensions from the cell wall of the prokaryotic cell. What are pili? Tubular extensions. They have the protein called as what? Pillin protein. They have what protein, ma? Pillin protein. Fimbriae are bristle-like structures sprouting out of the cell membrane. Remember these two descriptions about the non-motile structures. Pili are what? Tubular extensions. These are what? Bristle-like structures sprouting out. What is the function of pili? Attachment to the substratum and promoting conjugation between the two bacteria, that is promoting sexual recombination between the bacteria. Fine. Next, when it comes to fimbriae, bristle-like structures sprouting out, I have told you, no, this are only function is to attach to the substratum. The only function of fimbriae is what, ma? Attachment to the substratum. Clearly about the metabolism, that is nutrition and respiration of bacteria and reproduction of bacteria, we are going to learn in biological classification. So, with respect to the complete cell structure of prokaryotic cell, you should be clearly aware of what? Cell envelope and plasma membrane extensions, organelles present in the cytoplasm, genomic and non-genomic DNA, and then the motile and non-motile structures present in the prokaryotic cell. Fine? Good. Is it completely clear about the cell, ma? Fine. Prokaryotic cell is clear to your mind, no? Now, what we are going to learn is the differences between eukaryotic cell and prokaryotic cell. Then we are going to have a brief discussion on plant cell and animal cell. Then we will practice the objective as well. So, now shapes of the cells. What is the size of the eukaryotic cell? 10 to 20 micrometers. Few times only from this area you got matched the following question in the NEET exam. So, remember that. Typical bacteria has 1 to 2 micrometers. PPLO, pleuropneumonia like organisms, just 0.1 micrometers, and viruses 0.02 to 0.2 micrometers. Remember all the sizes of different cells. Now, coming to prokaryotic and eukaryotic cell beta. In eukaryotic cell, the outer limiting boundary, as which scientist said about the limiting boundaries of plant cell and animal cell? Schwann. Schwann, very good. It is Schwann, that is British zoologist, right? So, he said about the limiting boundary of a eukaryotic cell. If it is plant cell, what is that? Cell wall. If it is animal cell, cell membrane. But in a prokaryotic cell, you have what? Cell envelope. Okay, that is the first difference. Prokaryotic cell has what? Cell envelope. Second, we have to discuss about non-membrane bound organelles. First, what is that, ma? Non-membrane bound organelles. In eukaryotic cells, if you have to consider animal cell, you have two non-membrane bound organelles, they are centrioles and ribosomes. In plant cell, non-membrane bound organelle is only ribosome, whereas the non-membrane bound organelles in prokaryotic cells include ribosomes and inclusion bodies. What are they, ma? Inclusion bodies. Inclusion bodies in a prokaryotic cell are nothing but the storage bodies which are present inside the cell. Okay? Storage bodies like synophysin granules, glycogen granules, phosphate granules, sulfur granules, these are what? Inclusion bodies. These are also non-membrane bound. Coming to the ribosomal type, prokaryotes have only what? 70S type. Eukaryotes will have only 80S type? Very good. 70S you will see in chloroplast and mitochondria. Okay? I will discuss with you now. Ribosomes you have both 70S and 80S type. 70S you will find in the cell organelles like chloroplast and mitochondria. I will tell you in detail, do not worry. Next, when it comes to membrane bound organelles, do you see any membrane bound organelles in prokaryotic cell? No, there are no membrane bound organelles. But in eukaryotic cell, you have single membrane bound organelles and double membrane bound organelles as well. We will discuss it now. Now, next point is about the DNA. Okay? In eukaryotic cells, you have only genomic DNA in the cytoplasm and semi-autonomous DNA or organelles are present in chloroplast and mitochondria. Whereas, inside the cytoplasm of prokaryotic cell, you have both genomic DNA and non-genomic or plasmid DNA. Fine. So, these are the primary differences between prokaryotic and eukaryotic cell. So, one trick I will tell you beta, whenever you are looking into any cell structure, be it prokaryotic cell or eukaryotic cell, come from top to bottom, from the cell envelope, outer surface to inner surface, that is nucleoid in the prokaryote, nucleus in eukaryote. Last point, if you have to discuss about cell division. Cell division in prokaryotic cell is more rapid than eukaryotic cells. Fine. Now, first we will discuss about plant cell. Now, as I have told you, we have to come from the outer layer to the inner layer. First is what? Cell wall. Cell wall in a plant cell is composed of what, Nana? Cellulose, cellulose, hemicellulose and pectin. It is composed of cellulose, hemicellulose and pectin. Fine. And if you have to see the cell wall of a plant cell, at the time of division when two cells are dividing, in between two plant cells, middle lamella is formed. What is formed? Middle lamella is formed. That is how two cells are divided first, right? Like this, two cells are divided. Middle lamella has calcium pectate and magnesium pectate that is secreted from Golgi bodies. Next, inner to the middle lamella, primary cell wall is formed. What is formed, ma? Primary cell wall. Inner to primary cell wall, secondary cell wall is formed. And this is the cell membrane, cytoplasm. Fine. So, now if you have to see organization, first will be your middle lamella followed by primary cell wall, followed by secondary cell wall, followed by the cell membrane. Fine. This organization is important. Middle lamella is composed of what? Calcium pectate and magnesium pectate. This is about the cell wall of a plant cell. Clear, right? Now, next cell membrane, that is fluid mosaic model, we will discuss when we are discussing the chapter in detail. Coming to non-membrane bound organelles, only non-membrane bound organelle you will see is what? Ribosome. Okay, see, ribosomes are the only non-membrane bound organelles. Ribosomes, if it is in cytoplasm, these are 80S. Larger subunit is what in 80S type of ribosomes? 60S. Smaller subunit is 40S. In 70S type of ribosomes, okay, 70S type of ribosomes, what are the larger subunit? 50S. Smaller subunit 30S. That is it. Simple, right? This 70S type of ribosome is present where? In the chloroplast and mitochondria of a plant cell. Fine. Coming to single membrane bound organelles. What are the single membrane bound organelles that you will see? First is the endoplasmic reticulum. Endoplasmic reticulum is of two types. What are they, ma? RER, rough endoplasmic reticulum, smooth endoplasmic reticulum. RER will synthesize what? Proteins. SER will synthesize lipids. That's it. Second is what? Golgi bodies. Golgi bodies are single membrane bound organelles. What is their function? Packaging or synthesis of glycolipids and glycoproteins. Next, after Golgi bodies, what do you see? Lysosomes. Okay, lysosomes whose pH is very low and it is rich in hydrolytic enzymes. Fine. Next, after that, vacuole. 90% of the plant cell volume is occupied by what? Vacuole. Vacuole is surrounded by single membrane. What is that? Tonoplast. Vacuole is surrounded by single membrane. What is that, Nana? Tonoplast. So, this is about vacuole. Fine. Now, here you have plant cell wall is very thick, right? How the molecules will pass between two cells, how the transfer of molecule will take place? These are through plasmodesmatal connections. These are through what? Plasmodesmatal connections. Next, the cytoskeleton, which includes microtubules, microfilaments. Now, the double membrane bound organelles. Double membrane bound organelles include chloroplast, mitochondria and nucleus. These are the double membrane bound organelles. Clear to you, ma? Fine. So, what are the non-membrane bound organelles in the prokaryotic cell? Only one. What is that? Ribosomes. Single membrane bound organelles? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. And next is what? Double membrane bound organelles, nucleus, mitochondria and chloroplast. Clear with the plant cell? You got a clear idea about plant cell? Once clearly look into the labelings, because we have diagram based question on this in the coming slides. Venkatesh, I said you that I will reply to you at the end of the class, ma. Anything relevant to concept you ask, I will answer you in between, but this is a generalized question. I will ask you at the end of the class. I will answer you at the end of the class. Next. Now, animal cell. Fine. So, yes, animal cell limiting boundary is what? Cell membrane again, and here you have microvilli, means which cell is it? The cell from small intestine, right? So, it has microvilli and plasma membrane as the limiting boundary. Same story again. Single membrane bound organelles are what? Endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum will have ribosomes, extensions of nucleus, right? They will synthesize proteins. Smooth endoplasmic reticulum only in animal cell, along with lipids, they will also synthesize steroids. What do they synthesize? Steroids. Fine. Next is about what is the second single membrane bound organelle, that is Golgi bodies. Same case, that is packaging, fine, or synthesis of glycolipids and glycoproteins. And then is lysosomes, same thing, rich in the hydrolytic enzymes. Name few hydrolytic enzymes, ma, which are present in lysosomes. Lipases, carbohydrases, hydrolases, very good, proteases. These are the hydrolytic enzymes which are present. Next, after the lysosomes, it is vacuole, that is very less when compared to the plant cell in an animal cell. Next, here non-membrane bound organelles are two in animal cell, as we have discussed. What are they? Centrioles and ribosomes. Fine. Ribosomes, same case, ma, you can find both 80S and 70S. 80S you will find where? In the cytoplasm, and 70S you find in the mitochondria, because there is no chloroplast in an animal cell, right? 70S you will find in the mitochondria. Next, centrioles. These centrioles are present inside the centrosome. Okay? Centrioles exist as two cylindrical structures which are perpendicular to each other, like this. fine. And if you see, if you transfer sect it and see, you can observe a cartwheel arrangement of 9 plus 0 arrangement, where at the center you will have proteinaceous hub, attaching to it, it has 9 radial spokes like this. Each radial spoke will have 3 microtubules. This cartwheel structure we will discuss in detail when we are starting our classes. Fine. So, about centrioles, it is non-membrane bound organelles. They are present as 2 cylindrical structures perpendicular to each other, and they are in 9 plus 0 arrangement. Clear? Now, so this is about ribosomes and centrioles, non-membrane bound organelles in animal cell. Are centrioles present in a plant cell? No. Then how does the cell division activities are done in a plant cell? It has microtubular organizing complex, okay, which helps in the formation of spindle apparatus and promotes cell division. But here centrioles in animal cell, what they do ma? Cell division and spindle apparatus formation. Next, double membrane bound organelles here are 2. What are they? Nucleus and mitochondria. Those are the double membrane bound organelles. Got the complete picture on animal cell? Just in a fraction look into all the labelings properly. Done right now? Good. Now, let us master this cell, whatever we have discussed. We have covered from the official syllabus of your Cell the Unit of Life in NEET. We have covered 60% of that today. Fine. So, now if we have to see the questions on it, like 50 to 60% is covered. First, about discoveries you have to answer. It is Leeuwenhoek. What is the discovery of Leeuwenhoek? Which option? Leeuwenhoek has discovered living cell, right? So, which option? Very good. Robert Brown? Very good. Schleiden? Good. Schwann? Plant and animals. Both he observed. So, now tell me what is the right answer? B. Good, Nandu. Next, same. Diagram of plant cell is given. We have to select the option with current identification about the feature of the organelle. So, first we have to know what are the organelles that he has given. This is labeling what? Extensions of nuclear envelope, and here you can see ribosomes, means what organelle is it? Good, rough endoplasmic reticulum, right? And these are chloroplast structures. If you can remember the labeling of plant cell, this is what? Chloroplast structure. Fine. And this is what? 90% of cells volume is occupied. This is vacuole. And these are the plasmodesmatal connections. Now, look into the statements properly. B, is it smooth endoplasmic reticulum? No. So, you can cancel this statement. Next, D, it is not covered by double membrane. Good. It is covered by only single membrane. Next, C, because cristae you will find in where? Mitochondria. Good. Plasmodesmata is concerned with exchange of materials between plant cells. Done? Good. Next. Look into this. Here also you just label it. So, this is about nuclear envelope, right? So, S is nuclear envelope. And these are what? 2 perpendicular structures. Just now we have discussed centrioles, right? Good. And these are what? Golgi bodies. Good. This, very good, smooth endoplasmic reticulum. Now you answer. P, Golgi apparatus is mainly concerned with packaging of materials. This is correct one. Next, R is centriole and concerned with cell division in plants and animals. No, it is not concerned in plants. Next, S is the plasmodesmata and it is concerned with transportation of materials between cytoplasm and nucleoplasm. No, plasmodesmatal connections you see where in the plant cell, and here between nucleus and cytoplasm it is nuclear pore and nuclear envelope that mediates. Q is what, Nana? Smooth endoplasmic reticulum and is mainly concerned with synthesis of proteins. Wrong. In animal cells it synthesizes what? Lipids and steroids. This question you see, statement based question. It is about mesosomes, which is equivalent to which organelle of eukaryote? Mesosomes in prokaryotes is equivalent to which organelle of eukaryote? Anyone in the online platform, you can also answer, ma. Mitochondria, right? Mesosomes of prokaryotes are equivalent to mitochondria of eukaryote. Now you tell me, they help in what are the functions? Cell wall formation, respiration. Next, increases the surface area. True. Next, they are present both in pro and eu. Wrong, because they are present only in the prokaryotic cell, not in the eukaryotic cell. Now which statement? 1 and 2. Fine. Next, assertion and reason type of question. Description of pillae and fimbriae. Fimbriae, are they elongated tubular structures? No, pillae are elongated tubular structures. So this is wrong, and pillae do not play any important role in motility. This is true. Fimbriae are what? Bristle like structures sprouting out of the cell membrane. Fine. So this is true, reason is true and assertion is false. Next, which of the following are seen both in the prokaryotic cell and eukaryotic cell? Semi-fluid matrix called as cytoplasm. True, right? Next, selectively permeable membrane is present both in pro and eu. True. 70S ribosomes present both in prokaryotic cell and eukaryotic cell. Membrane bound organelles not present in prokaryotic cell. Next, genetic material DNA is present. Yes, which type of DNA he did not give, no. If he is giving linear, then we can eliminate, or circular only we can eliminate. But he said genetic material DNA, it is present. Chromatophores not present in eukaryotic cells. Now you tell me what is the right answer? A, B, C and E. Okay. Next, this is your NEET 2025 question, just from today's class. Now you answer, Nandu, you will get confidence. Yours, Aisha. We will look into the options first, okay na? The eukaryotic ribosomes are 80S, prokaryotic 70S, correct only, no? Each ribosome has 2 subunits, correct? The 2 subunits of 80S ribosomes are 60S and 40S, correct? While that of 70S are 50S and 30S, true? Next, the 2 subunits of 80S are 60 and 20, wrong. The 2 subunits of 80S are 60S and 30S, wrong. Okay. So, what options we have to go through? A, B and C. C, right? Look into that properly, all the A, B, C 3 statements. Right, no? D and E are wrong because the subunits given are wrong, fine. So, that is how A, B and C. So, with this what you have understood, it is not the amount of content you gain. You know the content properly. Whatever we are discussing, we should understand the concept and we should apply it in the objective. Application is the important segment here, fine. Be it a statement based question, match the following question, diagram based question, assertion reason question, whatever the question it might be, we have to apply our knowledge in a proper way. That application is the deciding factor for your NEET mark. So, this is a glimpse of this chapter. Tomorrow we will discuss on cell cycle and cell division as well. Thank you.